You are listening to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bandettini. Here, I interview neuroscientists and discuss their work as well as the latest developments, issues, and controversies in the field of brain mapping. Today, I'm talking with Dr. Alex Fornito, who has been a leader, educator, and innovator in brain imaging. A major emphasis of his work concerns understanding foundational principles of brain organization and their genetic basis, characterizing brain connectivity disturbances and psychiatric disorders such as schizophrenia, and mapping how brain networks dynamically reconfigure in response to changing task demands. He's currently the Sylvia and Charles Verdel Foundation Senior Research Fellow, Professor of Psychological Sciences, and head of the Brain Mapping and Modeling Research Program at the Turner Institute for Brain and Mental Health. He also leads his Neural Systems and Behavior Lab. He completed his clinical master's in neuropsychology and Ph.D. in 2007 in the departments of psychiatry and psychology at the University of Melbourne before undertaking postdoctoral training in the Department of Psychiatry at the University of Cambridge under the auspices of the NHMRC Training Fellowship. And there he worked with Ed Bullmore. He's also co-authored a truly outstanding book, along with Andrew Zaleski and Ed Bullmore, on network neuroscience titled Fundamentals of Brain Network Analysis. His collaborators are all over Europe, the US, and Canada, and he's an active member of OHBM, where he has recently been quite instrumental in establishing the virtual platform for the meeting. So in this podcast, we have a pretty wide-ranging discussion, starting with just talking about his book a little bit, and then going into talking about his pre-processing work, specifically trying to address, and I think making really good progress in addressing this problem of physiologic noise and how to get rid of it. And then we go into his main area, which is connectomics, looking at the brain as a network, doing network modeling. And then we go into his work sort of characterizing that, and also looking at how they change with activation, how they change with disorders, the genetic correlates of these network properties as well that seem to show up in terms of, especially in terms of correlates with the hubs themselves, which are very different than the other areas in the sense that they have a higher metabolism, they have slower hemodynamic or slower fMRI measured dynamics as well. So that's the main part. Then we talked about some of the limits of just understanding disorders with network connectivity approaches. And one interesting insight there was that disorders travel along these networks, and either the networks show pathology or the brain compensates, or a little bit of both. Maybe there's compensation and then pathology. So we talk a little bit about that. We talk about some of his thoughts on the future of connectomics and how it might make a clinical impact. Lastly, we wrap up with talking about his role in helping the OHBM, the second iteration of the OHBM virtual meeting. Last year, it was done very well, but done very quickly. And there were some changes that probably were a good idea to make. And he was instrumental in sort of spearheading a lot of these changes. And you'll have a little bit of a preview as to what the virtual setup might look like there. Virtual meetings in general are completely challenging, especially for international meetings of this size. And I think he's done an amazing job sort of preparing this. So, and also virtual meetings might be here to stay. Even if we go back to meetings, there might be hybrids. So we talk a little bit about that as well. So enjoy the podcast. Thank you. Okay. Alex Fornito, thanks for coming on to this podcast again. You're my first second time visitor. Very different context. This time we're talking about your work and your contributions to OHBM, which we'll get to at the end with the virtual meeting. But I'd just like to, I've been looking over your papers, and you've been very active in the field since about 2008, and you've been, your trajectory in terms of publications and whatnot has been, completely upward, and your impact has been huge. And I'd just like to ask, so how did you get started in this? What made you interested in this particular niche? Or was it just sort of something you sort of worked into? Like, what was your initial interest when you were younger?
Oh, thanks, Peter, and thanks for having me on again. It's a real pleasure, and I'm a big fan of the podcast already, so it's a real privilege. To be frank, when I was in high school, I was not really interested in science at all, and I think that was a product of being taught science in a very dry way. So I entered university wanting to be an author, funnily enough, and I was planning on being an English major, but after one semester, I realized that was not for me. And I'd taken psychology as a fallback and kind of got more interested in that, and was intent on forging a path working as a clinician. In Australia, at the end of our undergraduate degree, we do a year called Honours Year, where we do a small research project. And I sort of stumbled into a group led by Chris Pantelis, who was doing psychiatric imaging. And this was my first exposure to MRI, and I worked on a simple project using T1 weighted scans. And I just really loved it. It was a great team to work with, great environment. I loved the process. And so I thought, oh, well, maybe I'll kind of do some clinical training along with a Ph.D. And through that process, I just got more engaged in my research, and really got heavily into neuroimaging, the methods, thinking about brain connectivity and networks, and never looked back since. So it's been a bit of a circuitous route, and I guess I've been playing catch up ever since, in a way.
Well, it definitely doesn't seem like you've been playing catch up, but and it's nice to see that. It seems that a lot of people actually, right, when they start out, they're not quite sure. They try things. It's, what you said, it's just worth it to just, to try different things. And usually, and always to be aware, and usually it ends up being a circuitous route. And that usually works out if you keep an open mind and you keep trying new things. And it's great that a lot of it's serendipity. You end up with a good group that's really supportive, and you go from there.
Absolutely.
Yeah. So, and it really does seem like, your group, and it seems like there's a certain network within Australia that's actually really, really good. And it seems like you have something really good going there overall. So speaking of writing, I just want to start out with talking about your book that you published a few years back, Fundamentals of Brain Network Analysis. And I just have to say it's super, at least from my opinion, from looking at it, it's really clearly written. And it's interesting. It's advertised in some places as a textbook, but it's much more readable than a textbook. It's sort of like you can just read it from cover to cover, and it's engaging, it's interesting, it conveys the cutting edge concepts, but it does it in a way that you feel like, oh, this just all makes sense. So definitely writing talent is going on there as well. So, and yeah, definitely in science, I think I have to emphasize that people don't realize how much writing they do in science, and I just have to congratulate you on that.
Yeah, thank you. I think our intention with that book was to try to provide an accessible introduction to network analysis techniques for people that don't have a background on the one side in that area, because there are some excellent network science textbooks, but that can be quite a steep learning curve for someone who doesn't have a background in physics or engineering or math. But on the flip side, also for people with those backgrounds to show how these techniques can be applied in a neuroscientific context. Because although network science is quite general and can be applied in lots of different domains, the applications do have a lot of domain-specific nuances. And so it was really trying to reach out to both of those audience, and I was really lucky to work with my co-authors, Andrew Zaleski and Ed Bullmore, who obviously really contributed a lot to try and get it into shape.
Yeah, I have to say also, I read Ed Bullmore's book a while back, The Inflamed Mind, I believe it's called. And once again, Ed is a talented writer as well. So I've struggled myself. I've written a tiny book recently on fMRI, just called fMRI and MIT Press. It's sort of like a small handbook thing, but I really struggled. And part of the reason why I struggled is because, it's one of those things that you just need to block out time to do it. And just before we get into the other discussions of things, what was your strategy for doing that? Because I'm always want to know about that. If anyone's writing a book, I mean, you need way more time, it seems, than what you think you need.
Yeah, I don't know if I had a strategy. It was a bit of a hectic time at that period. So, we'd had a schedule and we ended up falling behind time. And then it all came due at a period that coincided with the birth of my first child. And so maybe the solution was sleep deprivation. There were many days of sort of rocking the seat with one foot and typing with the hands and things like that. But yeah, I mean, I was just, as much as possible, it was really about trying to structure in extended, unbroken stretches of time, because I'm very similar. I find that I can't write a little bit for half an hour, then do something else, then come back to it. So it was really about trying to structure my diary in a way that gave me unbroken pieces of time to get through it.
Yeah. Well, and there's nothing like a deadline, right? Unfortunately, with my book, the editor kept on pushing back the deadline, saying, "Oh, you can get it done. Well, if you need more time," and so that was probably a bad thing.
Yeah, rookie mistake. You need someone who's going to hold you to account.
Right. And I think it makes for a better book. I think actually it causes more coherence in your writing. And like you said, these large blocks of time, I think, are critical.
I've always been a bit of a crammer, so yeah, if I've got a bit of a deadline, it's.
Well, that's great. That's great. And who knows? Maybe there'll be a second edition. I don't know if network neuroscience is evolving fast enough to add more on.
Oh yeah, it certainly has. There's been a lot of development since the book has been published, and we are talking about it. It's a matter of finding the time.
Yeah, exactly.
It is something that we would definitely like to do.
Okay, well, great. So what I was hoping to do today is talk a little bit about some of your work, mostly about the connectomics work that you did. But I have to mention that, I mean, you obviously do a lot of other work with structural MRI, with pre-processing and functional MRI processing as well. And I couldn't help but notice a recent paper that you just published, it's out in Neuroimage in 2020, on identifying and removing widespread signal deflections from fMRI data, rethinking the global signal regression problem. And this is sort of near and dear to my heart in the sense that it's been an ongoing question and I just recently had a small discussion with Michael Fox about what to do. Why do global regression? Why is it a problem? But even if you do that, and even if you try to use things like retro-ICOR, it's still really hard, I found, and I'm actually always puzzled over why retro-ICOR, this basically uses the regressor from respiration and sometimes cardiac, why it doesn't work as well as it does. And I think your work kind of hit upon why in some sense. I mean, why don't you just explain your approach? I mean, it was really nice. I think it starts with the carpet plots and how you ordered them relative to their correlation with global signal regression. And it really captures all kinds of other things. So why don't you explain it a little bit? Yeah.
Yeah, sure. I mean, I think this work was largely motivated or inspired really by some of Jonathan Power's work, showing that you do have these really pervasive signal changes that affect nearly the whole brain, and they're tied to variations in respiration. And so prior to that, I'd thought, well, GSR is probably a decent thing to do because the networks you obtain with it are a lot more sensible, keeping all the caveats with it in mind. But once we saw that, we thought, oh, wow, this is quite a problem. But we also thought, there's got to be a better way than GSR, right? And we were just playing around looking at the CARPA plots, and typically they are presented in a sort of pseudo-random order, like the voxels are listed along the rows and they're sort of pseudo-randomly ordered. If you order them in different ways, you can start to see a lot more interesting structure. And we found that you could either order the voxels according to their correlation to the global signal, or using some kind of hierarchical clustering. And we started to see that these widespread signal deflections were sometimes more complicated. So in some cases, you might get instances where half the brain is going one way, increasing signal, and half the brain is decreasing signal. And in those instances, GSR will be ineffective, right? Because the mean signal will be more or less zero. And so you won't be removing much. And we found that visibly these were sometimes linked to head motion. And so we thought at least some of these are probably related to noise. You can never be sure unless you've got really extensive recordings. The data we were looking at didn't. And so we thought about, how can we develop a method that might remove these, and what happens if we remove them. So we developed an approach based on a clustering technique called DB scan, which allows you to kind of pull out diffuse weakly connected clusters. And so that we sort of apply that based on signal similarity between voxels. That allows us to pull out these kind of representative signals of voxels that have got different types of signal fluctuations. And we found that this approach could pick out, signals that were clearly noise with quite strange time courses. So, early spikes and then a relatively flat trace, or, other kind of weird and wonderful things that GSR would not be able to pick up. So in the validation paper, we thought, okay, well, let's do this and let's see how far we go. Now, the implicit assumption in the method is that these widespread signal fluctuations are noise, right? It's not being, it's blind to whether there is a broad signal change that is signal or not. So we make that assumption and we thought, okay, well, how far can we go if we try to remove all these structures? Are we left with anything? And in that paper, we looked at 3 or 4 different data sets, and this was work led by Kevin Aquino and Ben Fulcher, who did a really thorough job. And we found that you had better denoising performance on a range of different quality control metrics. But we also had evidence that you had better statistical sensitivity for example, pulling out resting state networks using ICA or mapping task-related activations. So it suggested that removing these noise structures actually potentially improved your power. The question is, how far do you go? It's an iterative method, and the number of iterations is a free parameter. We just kind of choose an arbitrary number. I think it was either 3 or 5 in the original paper. And so ideally, we would be able to develop a principled way. We have found, even though that first analysis looked at 4 different data sets and the same parameters seem to work okay for all of those, in other data sets, we have found that that can be too aggressive. And so the real trick is in trying to work out how far you go. And that's kind of an open area.
Yeah, that's a little bit right. That last part, right? Too aggressive would mean what? Like you're reducing some of the actual signal in some sense. So,
yeah. You end up with very weak correlations, not much network structure.
Yeah, interesting. Yeah. Okay. So and yeah, that's actually still. I mean, it's interesting because it does seem like if one could remove all the physiologic fluctuations, suddenly the sensitivity would go up tremendously. But then, right, you get this point where there is this weird mixing between physiologic noise and actual, real neuronal related fluctuations, and then you have to.
It's a really challenging problem because sometimes those physiological variations will be actually tied to cognitively relevant events. And so there's always a concern that you're throwing the baby out with the bathwater. That said, if you just look at a non-denoised carpet plot, you see these huge signal changes, and they're just going to dominate everything. And so, finding the right middle ground, I think, is still a big challenge for the field.
Yeah, it's definitely. But I think this really pushes things a little bit more. And I think that, and it sort of further highlights that there's intersubject variability of even physiologic noise. I mean, where these patterns emerge, and so it's more like anything. It becomes more complicated than you first think. So, all right. Well, that was a nice piece of work that just recently came out. So, just switching gears completely. Well, not really completely, but switching gears to connectomics. It's your main focus. I've been following pretty closely the literature, and my group does a little bit of this work. I'm going to ask some sort of general high-end questions first, just to get a perspective. So, the idea, and this is really nicely sort of brought out at the beginning of your talks, and actually it's brought out by a lot of people, but still the whole concept that the brain is both, has segregation and integration. You have modules, and the modules have to be connected. And this balance between segregation and integration really is sort of hitting a sweet spot of energy and efficiency and other aspects. So the first question that comes to my mind is that, we're limited with fMRI to study the brain at a certain spatial scale. And it's not, it actually is great that it works out that these modules appear to be at the scale of fMRI. Does this concept, I mean, it seems like and even, it goes back to even Cajal, as you talked about, balancing space, time, and material. Does this concept of segregation and integration, how does it vary across spatial and temporal scale? I mean, for instance, if you went down to individual neurons, or, obviously you have columns, does it break down? Is it, and where it does, it seems like there's principles to be derived in some sense regarding that.
Yeah, I mean, I think that's one of the kind of major unresolved questions. So I guess, when you're asking about segregation, integration, I guess you're referring to the network architecture that can support those two functional properties. And so what we've learned with quite a large series of network neuroscience studies by people like Olaf Sporns and Martin van den Heuvel and Danny Bassett and their teams, is that brain networks are modular in that if we think of the brain as a collection of nodes or specialized regions, these nodes can be decomposed into subsets of nodes that are more highly connected with each other. And so we assume that they form some kind of functionally related system. And that supports segregation or specialization of function. But then each of these modules has one or more hubs. So hubs are highly connected nodes. And these hubs are strongly interconnected with each other. So they form like their own kind of hub module that overlaps with these more specialized modules. And that's what we call a rich club. The idea being that the hubs are rich in terms of the number of connections they have, and they're a club because they're tightly interconnected with each other. And this rich club really supports integration. It provides a really efficient way for disparate parts of the brain to communicate with each other. Now, we can very clearly and robustly see that with macroscale human brain imaging, but we've also seen it in mesoscale track tracing data in mouse, rat, and macaque. We see it at the level of individual neurons and synapses in C. elegans. people have characterized these properties using calcium imaging in mice, and you see it. You also see evidence for the emergence of rich clubs in vitro, right, in neuronal cultures. So it does seem like a fairly robust multi-scale phenomenon.
Yeah.
You know, the degree to which maybe you would see that organization, say, within a column or not, I don't think has been examined yet, but that would absolutely be an interesting question.
Yeah, and it seems to suggest, though, that and you talk about this as well, that there is a cost in being physically distant, neurons being, because otherwise you can imagine a completely homogeneous brain with all the neurons doing their own thing, and they're forming these clubs, but they're sort of virtual in some sense. But being physically close works better for maybe an energy standpoint or speed of processing standpoint, or even the likelihood. I mean, like you know. Maybe I'll jump ahead a little bit. You talk about genetic influences versus environmental influences on these on hubs versus the peripheral connections, and it seems that you suggest that there's a genetic, more of a the mapping out using the Allen Brain Atlas, the genetic correlates of, let's say, metabolic activity. It seems to form these hubs, and it seems to correlate with these hubs, which seems like there's a scaffolding that's built, and then that's formed by an environment. The lesser connections are sort of tuned by the environment in some regard. And does that summarize that pretty well? I don't.
Yeah. Look, absolutely. So there's a few things to unpack in there. So one is, we know that physical distance does play an important role in shaping brain connectivity. So generally, areas that are near each other in space are more likely to be connected and more strongly connected. There's roughly an exponential decay in the probability of any two areas being connected as a function of their distance. So it decays quite rapidly. And people have used this. So people have developed models where you can wire up a brain completely at random, but subject to this kind of distance penalty, and you will be able to grow a network that has many kind of complex properties that look like a brain. It has modules and all sorts of other features. Yeah. But it doesn't get you all the way there, and that's because the brain does have a few extra long-range connections that are beyond what would be predicted by that simple model. Okay. And these long-range connections tend to be concentrated on the hubs, this rich club, right? And that makes sense because these hubs are sort of integrating disparate parts of the network. You sort of think of the global air transportation network. You've often got connections between a hub in one country, a hub airport in one country, and a hub airport in another country. The brain has a sort of similar organization. What appears to be the case, if you look at C. elegans, where we know the birth time of each and every neuron, it turns out that all of the hub neurons are born quite early, so before the animal hatches, whereas other neurons continue to be born afterwards. So it suggests that they're established quite early. There is some evidence in mammals that areas that we might think are hubs might also develop earlier. So it does suggest, and there is a bit of evidence from preterm infants supporting that idea as well. So there is a kind of interesting idea that perhaps these long-range connections between hubs are established quite early and form an initial scaffold to which other parts of the brain kind of connect up. But these connections also have quite a lot of plasticity. I mean, we've done work, there's been a lot of other people that have done work, developmentally, we know that these long-range connections between hubs, which tend to be association areas, have this protracted period of development into late adolescence, early adulthood. And so in recent work, it's on bioRxiv at the moment. It's will hopefully be in print soon. This is led by Arina Adnatkevičiūtė, who was a PhD student in our lab at the time. We sort of asked, well, given these properties of hub connections, is there something genetically special about them? And so we started with the twin data from the HCP, and we just used standard heritability modeling to understand genetic influences on variations in connectivity strength. And so we did this across the entire connectome. And then we asked, well, is heritability on average higher for those rich links between hubs versus other type of connections? Yeah. We found this was indeed the case. And so it suggests that genetic influences on phenotypic variance in connectivity strength are not distributed homogeneously throughout the brain, but they're really concentrated on those kind of costly but really functionally valuable connections between hubs. Now, the critical thing here is, the caveat is that it's genetic influences on phenotypic variance. So we're really looking at individual differences. So it doesn't mean that genes are not important for connections between non-hubs. And, we certainly know that early in development, there are, very strong, robust genetic patterning centers that are established in primary sensory areas that are non-hubs.
Yeah.
But what it does suggest is perhaps those are quite conserved, and, the variance between people in those connections are not really driven by genes, but more so environment. So they're quite plastic. Whereas the connectivity between these between hubs is to a large extent driven by genes.
Well, that's interesting.
So individual differences in the connectivity.
Yeah, the individual differences in connectivity are driven by the genes and the hubs themselves, as opposed to, that's interesting. And I all right, okay. And is there anything that characterizes the hubs in terms of, you were mentioning in one of your papers about, certainly they're metabolically more active. It's metabolically more costly to have these. You can imagine the hubs have, these multiple connections coming in. Is there like a higher density of neurons? Is there somehow the structure, is there any differences as well?
Yeah, so, this is a really hard question to tackle, because you're really trying to link up macro scale and micro scale property, and it's hard to kind of access those data. A lot of really nice work on this front has been done by Martijn van den Heuvel, and his team has presented some evidence to suggest that the neurons in hub areas have got, particularly the pyramidal neurons, have got a greater dendritic complexity and a larger somal size. And there's some evidence to suggest that areas that we might think are hubs have lower neuronal density. So it's not so much the number of neurons, but more the synaptic connectivity of those neurons. Okay. And that makes sense because we know, metabolic signals that we pick up with imaging are largely related to synaptic activity. And so if there's a lot of synapses in there, and we expect them to be highly active because these hubs are really central elements of the network, then we suspect that's what probably is driving it.
So one other characteristic that you also talk about with hubs, I'm going to go into also with disorders as well, but and this struck me as kind of counterintuitive, that looking at the connectivity strength is higher, but the frequency content, if I understood it correctly, the frequency content is generally lower than these peripheral. So if you look at connectivity between hubs or within hubs with other areas, it's lower frequency content and higher frequency content in peripheral connections in that sense. So to me, I'm looking at a hub and I'm thinking, oh, it's always turning on and off because it's sort of like a switchboard in some sense. But in some ways, you're saying maybe the long range connections are slower in some sense. How would you explain that?
Yeah, I mean, it kind of ties to really a growing body of work looking at regional timescales of activity, and a lot of this started with some really nice findings published by John Murray in, I believe it was the macaque, looking at electrophysiological data, where if you look at different regions along the visual hierarchy, it seems that areas that are higher up in the hierarchy have slower intrinsic timescales, just in terms of the autocorrelation of each region's firing patterns.
Okay.
Now, to a first approximation, the hubs of the brain tend to be areas residing in transmodal, paralimbic, multimodal association cortices. So typically areas we would think of as being near the top of the cortical hierarchy. There's some subtleties there, but generally they tend to be the hubs. And so, given what we see in the mouse, we would expect to see a similar pattern. There has been quite a few studies recently doing similar kind of timescale analyses showing that the timescale tracks what we think of as cortical hierarchies. And in our own work, we started with, this was work led by Ben Fulcher and Sarab Sethi, where we took data from the mouse. We used the Allen Institute connectome based on track tracing. And then we had fMRI data. This was a collaboration with Valerio Zerbi. And we found that areas with higher in degree, so more incoming connections, tended to show greater low frequency power in the bold signal. And this was, this was a kind of really big data trawl where we computed something over 6,000 different measures of univariate time series properties. And, this was kind of the most robust effect that came out, trying to relate these properties to structure. So it aligns with this idea, and it suggests that regions with lots of inputs have got slower timescales, and we presume that slower timescale of activity allows them to integrate information over broader time frames.
Okay.
And it aligns with the idea that these areas are involved in more complex processing that's not so stimulus driven or stimulus bound. We've also, Ben in follow-up work has also found a similar association in human data. So.
Okay. That's all right. That's actually a really good point to bring out, right? That it might be, more different types of calculations altogether that have slower timescales. That's so yeah. So if anything, right? I mean, in general, the concept of as opposed to starting out with with early fMRI looking at just areas of activation. Now they're connected by networks, and now there's hubs that seem like they are communicating with other networks through those hubs. And so now just shifting gears a little bit with pathologies. So obviously the bottom line is to not only to understand the brain and maybe derive principles about how the brain is organized based on this, but also to use this to characterize pathology of some way. And one thing that struck me right away, you were mentioning that I think in your really nice review article, Connectomics of Brain Disorders in Nature Review Neuroscience in 2015, that the brain, when given an insult, it's either maladaptive or adaptive, and and also that I was actually intrigued by the concept that the insult or sort of projects onto these networks and travels through the network, and then affects everything else through the network. And then usually you mentioned that it's manifest in the hubs as well. But my feeling is that if it maybe the behavioral effect doesn't show up until it gets to the hub in some regard. But who knows? But I'm wondering, what are the mechanisms? So you talk about the mechanisms. I was intrigued by this. That, I mean, you have certainly synaptic or neuronal connectivity, but you have all kinds of other transport along these networks. And it's interesting what would be those mechanisms for, if you have an insult somewhere to transport that disorder along that network.
Yeah, so I mean, it's a good question, and there's a lot of potential different mechanisms. We sort of talked about a few examples, and that wasn't in that paper, and that wasn't by any means an exhaustive list, but it does depend on the insult. So, in the case of say a stroke, a focal stroke, where you've got a localized lesion that affects the function of one area. Obviously, the brain operates much like a symphony, where the activity of one area depends on another, and so if one of the members of the orchestra is not doing their job, it's going to send other connected members out of sync. And this phenomenon was described over 100 years ago by von Monachov, the idea of diaskesis, and it's in a dynamic sense. You can compare that to a case of neurodegeneration where you have misfolded proteins that accumulate in the cells of one brain area, and they can then travel along axons through axonal transport mechanisms and get released out into synapses and then affect other neurons, leading to transneuronal spreading of disease. And this is something that has really become clear in the last 5 to 10 years, that this is occurring in a lot of different neurodegenerative disorders. So it does depend on what the underlying pathological mechanism is. But the net effect is that ultimately no single brain region is an island unto itself, and if you have dysfunction in one part of the system, it will spread to other areas.
Yeah, and I was even intrigued by you're mentioning the concept of hyperactivation, stressing the neurons and causing a certain amount of degeneration as well. Then one might think, oh, what is hyperactivation in that sense? Is it somehow this continuous firing as in seizures, or it's all these things I think are on the edge of not really being fully understood, I imagine. So.
Yeah, it is a tough question. We have this kind of, I guess at least as far as imaging goes, we just define it in a statistical sense, right? If you've got a group where there's been some kind of stress and you've got a control group, if it's if activation is statistically greater in the former group, then you assume there's hyperactivation. And then that could either represent some kind of pathological change or some kind of adaptive change that's trying to compensate for an underlying deficit. There is evidence that even if it is adaptive or some kind of compensation response, that over time it can turn into a maladaptive process where you start to see degeneration. And that is potentially because if the neurons in that area are kind of pushing their metabolic limit, eventually over an extended period of time, they'll start to be subject to various oxidative mechanisms that will increase their vulnerability to degeneration. Yeah. But I think really having a very clear definition of what abnormal hyperactivation is and whether that is a sort of transient adaptive response or more of a stable long-term maladaptive response, it's very hard to get a very specific way of characterizing that.
Yeah, no, I that's my sense as well from reading this. It seems that there's a description of either like for instance, sometimes your brain compensates perfectly, or sometimes it just starts to degrade, and it all depends on the pathology and the mechanisms of the pathology, it seems. And right, Alzheimer's mechanisms especially, it seems that's an example of this sort of thing. Or even even you've been looking at you have a recent paper in medRxiv looking at changes in functional connectivity with antipsychotic treated and antipsychotic native patients, just simply showing this sort of disconnectivity with psychosis, essentially, changes in connectivity. And it's important to bring out that looking at connectivity changes across these groups is extremely sensitive, and you can see these differences, and they sort of speak to trying to get some sort of traction in psychiatry in general of what are the mechanisms for this. Is there something about the specifics of the connectivity disconnections that speak to some sort of mechanism of what's going on in terms of psychosis?
Yeah, look, that's a really good question. Again, so I think we're nearing the end of the first wave of studies where we've developed these connectivity mapping tools. Let's use them to understand where the changes are. So we get these maps. And then the question is, well, what does it mean and how is it useful? So, from these maps, we've sort of realized that you can see connectivity changes from very early in the illness, and they're often quite widespread, at least particularly with respect to functional connectivity. With some evidence that certain networks might be preferentially impacted, such as corticostriatal systems, default mode, and perhaps some limbic areas. I think the question is, well, how did they arise? Well, there's two questions. How did they arise, and what are they useful for? So can these changes predict anything? Now, prediction is hard. Typically to do that robustly, you need large samples, and that's really hard to do in psychosis research. In terms of how we got there, I think the interesting question, returning to this idea of network spread, is, is this a disorder that is intrinsically got dysfunction that emerges in multiple regions or property of multiple connected systems, or is there something that occurs focally that rapidly spreads to affect other areas? We've done a lot of work in psychosis focused on corticostriatal systems. There's a lot of evidence to say that increased dopamine, particularly in subcortical areas, plays a really important role in the genesis of psychotic symptoms. And we have done a range of studies looking at the functional connectivity of these systems, showing that they're altered across a wide spectrum of illness severity, from high risk through to people with chronic illness. And more recently, this is work led by Christina Sabarodin, who was a Ph.D. student in our lab. We've tried to use DCM to get models of effective connectivity to try to work out, could we identify areas where perhaps, there's a really high out degree that would imply that, this is an area where the outgoing connectivity is disrupted, and, that would imply that area is perhaps a target. Yeah. What we found in the early stages of psychosis, when people are experiencing their first episode, and this is before they're exposed to antipsychotics, we do see quite prominent evidence of subcortical disconnectivity. Particularly in the striatum and midbrain. And so this is telling us that, perhaps early on in the illness, a lot of the pathological changes are occurring subcortically, and then perhaps they involve cortical systems down the track. So really we're kind of in this phase now of moving beyond these general maps and trying to home in on, what are the origins of these changes? And there's still a very long way to go, but that's the way we're tackling the problem at the moment.
Yeah, that's and obviously, people have to understand, right? There's huge benefit in doing group studies to try to understand these mechanisms of these changes, so that obviously the long range goal. I mean, people always are still right now. They're starting to talk about can you do this on individual subjects? Can you actually map the connectivity, and do you have enough sensitivity? Are there biomarkers for susceptibility for psychosis? And even if you did know exactly what was going on, are there any interventions such as TMS or deep brain stimulation or something like that might treat this in a more direct way than medication? Yeah. So yeah, there's all kinds of things.
Yeah, look, I mean, TMS is really an active area, and the results, in psychosis are not as clear cut as, say, in depression, but there is some promising signs. And I think, I enjoyed listening to your interview with Michael Fox on an earlier podcast, with his wonderful work using connectivity to find stimulation targets.
Right.
I think we need to develop more refined targets for people with psychosis, and perhaps TMS will work. Non-invasive brain stimulation also in theory holds promise, but is also a challenging area because there is certainly a very negative public perception of psychosurgery for historical reasons.
Yep.
So I think if we were to be able to get a robust evidence base to support that approach, then absolutely, there could be something to gain from it.
Yeah, that's, I mean, either way, right, having a better understanding of this and maybe a way of detecting it earlier, who knows? I mean, there's all kinds of interventions that are possible. And yeah, the ultimate goal, right, is to have, more traction in psychiatry in general, and to try to, clearly neuroimaging is lending these insights, even though, right, there's no treatments yet that are perfect. It's at least guiding this. So, so along these lines, once again, you mentioned, just really before we shift gears completely, where do you see your connectomic work going? I mean, we sort of hinted at this a little bit in terms of, maybe suggesting treatment, maybe better understanding different nuances and differences in disorders. What are the hard limits? Where are the opportunities? Any long-range perspective?
It's if you ever want to be wrong about something, make predictions about the future. So, I mean, I think if I'm going to go out on a limb, look, historically we've really focused on, I guess, developing and refining techniques for network mapping and measuring different network properties. That's become fairly mature. I mean, there's still work to do, and we'll continue to work in that space. But for us, the next step is trying to turn these measures into some kind of mechanistic insight. And so over the next few years, we'll be working a lot more with generative models of networks. So this is quite an exciting area where you can grow networks computationally according to specific wiring rules and see if you end up with something that looks like a brain. There's been some really promising work on this front, led by people like Rick Betzel and Petra Vertej. And we're starting to play with these, and also linking the connectome models to models of dynamics. So we've been working with Gustavo Deco and understanding the properties of neural mass models and how they might be constrained to generate better models of dynamics. So I think, it's really about trying to use these models to generate actionable mechanistic insights. And in parallel, I think we will continue to try to integrate genetics with the imaging measures, because the genetic data does give us, again, a different way of understanding the molecular basis of some of the signals that we're measuring. And it really is a bit of I mean, you mentioned opportunities. I think there is a lot of opportunity in this space at the moment, because now with large databases such as the UK Biobank, it's possible for almost anyone to do genome-wide association studies of particular phenotypes. We've got amazing resources like the Allen Institute Gene Expression Atlas, and of course, large-scale twin studies such as the HCP. So there's really a lot of scope to tackle the genetic basis of brain networks from multiple different angles.
Yeah. Okay. Well, that's I'm sure that there's nothing in that answer that I sense would be wrong at all. So
I hope you're right.
Sort of paints a picture that I think is actually really, there's a lot more work to be done, and it's a mature field that there's even people who still doubt the utility of even using connectivity as a measure but it's clear, obviously from all your work and all the work of many other people that while we still don't completely understand, we don't completely understand the nature of fMRI connectivity, it shows us really clear, consistent, repeatable correlates to many things. So it's and right, having these, as these databases grow, I think that the insights will continue. So, and you're right, the generative models, I do sense that there's, right, if you have certain constraints, and you could derive, you could start to build brains from bottom up in some regard, not like the, trying to sort of start with absolute first principles, but having these, starting with sort of these, right, as you, ideas of segregation, integration, and even the question of computation. Why does the motor cortex have to be as large as it has to be, or in terms of number of neurons or whatever? And there's a hub there in some regard, and there's a principle there, and yeah, it seems that there's all kinds of room for things like that, which may or may not touch on generative models or not, but still, I think that's great. Okay, so in the remaining time, let's just shift gears completely. And, I think that this podcast will be out before OHBM, the Organization for Human Brain Mapping meeting that's coming up is the 2021 one. It's the second virtual meeting. And the first one was great, except there was a lot that, was done on the fly, and it worked. And there's a lot maybe that we thought could be improved. And you've been completely instrumental as far as with OHBM in helping to create the second version of or organize the second version of the virtual platform. Could you talk about that a little bit? How it's going to be improved, and what to expect?
Yeah, sure. Look, it's been an exciting ride, and we're really getting to the pointy end now. So it's getting quite busy, but we're really excited about this new platform. Look, I think you've sort of explained it quite well. I was certainly very impressed last year with how the conference came off, given the time constraints and the rush to adapt to COVID. But there was, and obviously not many people had done online conferences before. But there was still a lot of things that there was a lot of room for improvement. And so following the conference, Ina and Randy, as chair and chair-elect of OHBM, reached out and asked me if I'd be interested in trying to form a bit of a technology task force to work on a new platform. And so we managed to pull together a great group of people, from just people that self-nominated, as well as representatives of the different OHBM interest groups, the special interest groups, and so on. And we went through a process of looking at the feedback, because OHBM did send around surveys asking about the last year's platform, and talking about what are the things that we would like to see in an online meeting. And really the key theme that came out was something that really facilitates social interaction and live exchange. We wanted to just embrace the fact that a virtual meeting cannot just be an in-person meeting online. It's fundamentally different. People don't want to spend 8 hours just passively watching Zoom talks. And so the question was, how can we enable that? And so we went through quite a process of consulting different software companies and solutions. And, we found that some were excellent for certain things we liked, but didn't cover other items. And, there was always trade-offs involved. But in the end, we settled on a company called Sparkle, which in some respects was a bit of a risky choice, because they had really largely developed their platform to enable concerts and more general social functions. So, for example, they've done Burning Man online and things like that. But they just, they provide a really unique space and set of functions that really do emphasize the social aspects. Okay. And so we've been working really closely with them, and they had they were very open to developing something that would be tailored to our needs. So we've been working with them very closely in trying to get everything together. And I have to give a massive shout out to Daniel Margulies, who is program committee chair, but is also doing an amazing job getting all the content in there and helping the Sparkle team, get all the features online. And we think that it's going to be quite a nice conference. There's really nice poster functionality. There's nice social functionality. So we hope that it will be it will address a lot of people's concerns about last year's meeting. That's not to say that I'm sure we won't get everything perfect, and there will be room for improvement. But the nice thing is that Sparkle is open source, and so that allows us to open it up to the community to develop bespoke solutions or features.
Yeah.
And, moving forward, we can continue to improve the platform as we get more experience with it and feedback. It is quite a big job that none of us are expert in, and we're learning as we go. And so it will probably take a couple of stabs at it before we get very smooth and, something really polished. But I do think that this year's meeting, we're pretty excited to see how it turns out.
So yeah, I mean, I think that's right. The features for, last year's meeting, it's nice to have obviously you also have to deal with the time zone differences as well. And I, the ability to sort of search posters easily. And I don't know if you have GatherTown, that was something that was last year. I mean, the idea of, that was for social events, but the idea of having something, maybe you will, with regard to just browsing posters and things like that could be really cool.
Yeah, I can give you a bit of a teaser. So there won't be, GatherTown won't be formally integrated within the platform. The poster hall will essentially come up as a list of thumbnails of all the posters that people can search through. And when you click on a poster, you enter a room, and people will, presenters will have had the option to upload a brief video that kind of walks people through their poster.
Oh, cool.
But there'll also be live times where the presenter will be, quote unquote, at their poster. And then you kind of enter this room and your video comes up, and you can have a video chat with the presenter, and other people can jump in as well. And there's also a kind of text-based chat. So each poster has its own kind of space, essentially.
Well, that's awesome. That's awesome. And yeah, no, okay. I'm looking forward to that. Definitely. And actually even thinking forward in the future, one thing this COVID has done, forcing virtual conferences, is the thought that there are certain things maybe virtual conferences do better. And also they allow for people who can't afford traveling to, and it would be nice to continue this in some sort of hybrid fashion. It's hard to know what that would look like exactly, but I think that's a general sentiment, that people would like to have a hybrid as well in the future.
Yeah, my feeling is that a lot of these changes will be with us for quite some time. So, I'm definitely looking forward to the day when we return to in-person meetings, although I'm in Australia and I don't think we'll be able to leave the country for a very long time. I think that there will be an online element with it, and what that looks like, I'm not sure. That will be for council to decide as we evolve and get more experience with the platform. But I think it will really help to increase accessibility of the meeting for a lot of people.
Yeah. Well, great. I'm looking forward to it. And thanks for spending all the time and your expertise to help work on that as well. And right, a shout out to Daniel Margulies, who's been chairing the program committee and working on this aspect as well. But yeah, all right. Well, thank you very much. I mean, this is not only a great perspective of your work and your book, but also kind of a perspective of, we've been talking to a number of people who are doing sort of network approaches to fMRI, and each one is unique, and they all are both insightful and hopeful in terms of what you can do. So, and it's always good to hear. So, yeah, well, thank you very much for coming on, and look forward to seeing your work in the future, maybe at OHBM as well. So,
yeah, thank you. It's been a lot of fun.
All right. Well, thanks.